Hi everyone, we are now entering week 5, so hopefully at this point in the course you're familiar with the type of activities that you're going to be doing each week. Now this week we're going to begin a study of a new case scenario. We're going to specifically look at cystic fibrosis, and the idea here with this one is that you will gain a better understanding of what our DNA molecules actually do. We know now exactly how they're constructed, what they're made of, and how they copy in themselves, but how do we take the information in that DNA molecule and actually turn that into a functional molecule that's going to carry out a range of activities inside of our cells? And it's a really important process, crucial that we get those working molecules at the very end of the process. If not, we could have something like a genetic disorder such as cystic fibrosis. So to understand this better, we have two main topics that we're going through this week. First off, we're going to look at biological molecules. We're going to talk about how important the structure of molecules actually is. And when I say structure of molecules, we're talking about the three-dimensional shape of molecules. Now, protein specifically is what's going to be involved when we're talking about cystic fibrosis, but it's absolutely crucial that our molecules maintain a perfect three-dimensional shape, and if they don't, then those proteins very likely won't function the way that we expect them to inside the cell. So that's one of our topics this week. You're going to have a lab that goes along with that, where you're really looking at shapes of proteins and how important that shape is to the protein's ultimate function. Then we're going to talk more about taking that information in the DNA and getting that protein at the very end. There's a couple of processes involved in that. We're going to have what's called transcription and translation, and we will have a lab activity that goes along with that one as well. And with that one, you're going to be working with those bead kits that you worked with last week, or really the last couple of weeks, to better simulate what's happening in the process of transcription and translation. So besides the lab activities, you also have a couple question sets that are going to be due this week. Everything again is due on Sunday night. If you have any questions at all on any of the activities, assignments, information that you're looking at throughout the week, definitely feel free to reach out to me. I'd be glad to answer those questions.